She's presenting for us today. And just before we begin, to orient everyone to our seminar platform, all attendees are going to be muted during the seminar. But if you have any questions during the seminar, please type them into the chat box, which you'll find on the GoToWebinar console, usually on the right-hand side of your screen. At the end of the seminar, we'll have time for additional questions. And at that time, we can also unmute people for verbal questions by using the raise your hand feature, and the question box will still also be available at that time. So as I said, today our speaker is Dr. Lisanne Petracca. She is a postdoctoral researcher within the Quantitative Conservation Lab and the Washington Cooperative Fish and Wildlife Research Unit, where she is collaborating with Dr. Sarah Converse, Dr. Beth Gardner, and partners at the Washington Department of Fish and Wildlife to assess gray wolf population dynamics in Washington State. She received her PhD from the State University of New York College of Environmental Science and Forestry, where she leveraged connections with Oxford University's Wildlife Conservation Research Unit and the nonprofit Big Cat Conservation Organization, Panthera, to study the spatial and behavioral ecology of large carnivores at the human-wildlife interface. She was employed by Panthera as a geospatial analyst and conservation scientist for 9+ years and is passionate about large-scale conservation, achieving outcomes that balance the needs of carnivores and people, and communicating complex science in a non-complex manner. So with that, I'll turn things over to our speaker, and welcome, Lisanne.
Thank you, Andy. I hope you can all hear me okay. This is my first time using GoToMeetings. I think we've worked it out. So thank you all for being here. I really appreciate your interest in my talk today. And yeah, so this talk is in general dedicated to all Indigenous peoples whose knowledge is so critical to the preservation of our natural systems and to the conservation of threatened species. This work that I will be discussing today is largely my dissertation work from SUNY ESF. And I'm really excited now to be working in the North American sphere on wolves in the Quantitative Conservation Lab. So I just started that position a few months ago. So I figured for this talk, I'd focus on what's in my wheelhouse. So here we go. My focus today will be on the use of traditional ecological knowledge in the quantitative toolkit for big cat conservation. So TEK is a pretty broad term. I like the definition used by the Fish and Wildlife Service, where, essentially it's described as the evolving knowledge acquired by Indigenous and local peoples over hundreds of thousands of years through direct contact with their environment. And this knowledge is specific to a location. It includes the relationships between plants, animals, natural phenomena, and landscapes. And, this knowledge is used in all aspects of life, including hunting, fishing, trapping, agriculture, and forestry. And it's really about the relationship of living beings, human and non-human, with one another and with the environment. So we'll be taking a whirlwind tour here. Sorry that this isn't an aquatic creature. I am a terrestrial wildlife scientist. So we'll start with the jaguars of Central America, and then we'll move on to a large mammal community in the largest contiguous protected area complex in any single African country. And this is a leopard, and this is within Angola, our study area for the second talk today. And then we'll conclude with the lions of Hwange National Park, Zimbabwe. And if that lion looks familiar to you, it is because, yes, that is the famous Cecil who was killed by a bow hunter in 2015. So the roadmap for today's talk. So yeah, I will discuss the integration of traditional ecological knowledge in assessments of single and multi-species occurrence, in the mitigation of human-carnivore conflict, and overall in guiding conservation and anti-poaching strategy. So this is a photo of me conducting a survey in the Tet province of Mozambique. So our first question today, this has been answered, turns out, with some work I did in the Journal of Applied Ecology. And the question is, can interviews with local residents serve as a reliable means of assessing jaguar presence outside protected areas in Central America? And so so the jaguar is the largest cat of the Americas. It is near threatened on the red list, and its range is from northern Mexico with, some individuals that do get across the border into the southwestern US, all the way to northern Argentina. Now, for a species that traverses such enormous range, in order to conserve the species, you need to think big. So I did work for about a decade with the nonprofit Panthera, which focuses on science-based approaches to conserve big cats. So their flagship program within the Jaguar program is the Jaguar Corridor Initiative, which, it's spearheaded by Panthera. It's in partnership with other government entities and nonprofit organizations. And it essentially envisions Jaguar range as these Jaguar conservation units, or these Jaguar strongholds, connected by a series of Jaguar corridors. And the idea is that the JCUs form the nodes of this connectivity network, with corridors serving as linkages through which dispersers can move. The focus of my talk today will be within Central America. So the big question was that we knew jaguars were in JCUs, these Jaguar conservation units. This is where there are established research programs, the areas are protected, there's intact forest, we know jaguars are there. But what about the corridors, and how to answer that question across such an enormous area? So keep in mind that some of your more traditional techniques, such as camera trapping, won't necessarily work. These corridor areas, they comprise community lands, private lands, a lot of agriculture. A camera exercise would require thousands of camera traps, and then you would need to get access to those lands, and then they might still be stolen. So we needed to find a different methodological approach. And so what we decided to do was to go with interviews with local residents. So I personally, with my team, covered the Jaguar corridors within Belize. And Belize, I don't know if any of you have been, but it has just an incredible wealth of ethnic diversity. There's a population of about 360,000 people. You have mestizos, who are the Spanish speakers. They're of mixed Spanish and Mayan descent. You have your Creoles, who speak Creole, largely of Western and Central African descent. You have your Maya, you have your Quechua Maya and your Mopan Maya, and the man on the left, he is a Quechua Maya. And you have your Garifuna people. So the man in the photo on the right is a Garifuna man, and they are of West Central African ancestry with Arawak and Island Carib thrown in. So there's just so many cultures, so many languages, and it's just a fascinating place to do field work. And so common among these different ethnic groups is a longstanding connection to the land and knowledge of wildlife and its sign. Many were raised in hunting families, and many continue to be subsistence hunters. And so in our interviews, this is a quantitative seminar, so I'll gloss over the content of the interviews, but we generally asked, how often each unit, and I'll discuss the spatial units in the next slide, how often they were visited in the last year, and then if that interviewee had seen tracks of jaguar and or the 6 main prey species of jaguar within the last year. And of course, we followed up with multiple lines of questioning to make sure that they were being truthful and as accurate as possible. So yeah, this is an example of our corridor areas here. So there were 12 corridors across 6 countries, and each corridor was broken up into grid cells. So using corridor 2 as an example, so I led field work in this particular corridor and in corridor 1. So each corridor was broken up into these grid cells. The sampling units were of fixed size, about 25 square kilometers, and the area represented, the area for which human subjects were expected to have knowledge about wildlife populations. They did tend to walk on average 5 kilometers in a single direction to and from their farms or on their hunting trips. And we also, and to reflect at the time, the average home range size of female jaguars, which was estimated on the smaller end back then. And so, yeah, we conducted 4 to 6 interviews in 60% of these units, and those are in dark yellow, and 1 to 2 interviews in the light yellow. And again, that number of interviews was set through simulations to determine, what level of precision that we'd like in our estimates. And so we sampled accordingly. These were the 6 prey species, starting from the top left and going clockwise. We have the white-lipped peccary, paca, armadillo, white-tailed deer, which likely is very familiar to you all, the red brocket deer, and the collared peccary. So with interview data, okay, we collected data, again, across 12 corridors, 6 years, 6 countries, within the same corridor. In some cases, we use different field teams. That is a lot of sampling heterogeneity over space and time, and we needed to account for that. As well, interview data are prone to false positives. Failing to account for false positives will bias your estimates high, which can be a really dangerous thing to do when you're working with a threatened species. These false positives aren't necessarily on purpose. They can be due to misplaced geographical accounts. They can be due to errors in recall timing. They can be due to species misidentifications. The identification that we were most worried about confusing were the mountain lion, or puma, and jaguar. And so we really worked hard to try to discriminate between the two. Yeah, so the important things to be aware of here. So to model site use, we used the false positive extension for single season, single species occupancy models by Royal and Link. And, essentially each sampling unit was treated as a site. So we have from 1 to R sites here, and each interview was a replicate at that site. So the total replicates at a site is T, the number of detections at a site is Y subscript I. And yeah, in the model itself, we have psi, which is our probability of habitat use. We have our P11, which is our probability of true detection, and P10, which is our probability of false detection. So if this looks like a bunch of gobbledygook here, what this means is that within any given sampling unit, and given a certain number of detections, or 0 detections, the species can be there, and the species is either detected or not on those occasions, or perhaps the species is not there, and it was either falsely detected, which is that P10 parameter, or it was not falsely detected. So again, you can, detect a species, you cannot detect it, you can also falsely detect it, and then, the opposite, not falsely detecting that species. So it's just accounting for the fact that a species can be there, but you fail to detect it or you falsely detect it. So our occasion for true detection probability, which is P11, we had that random intercept here of field season, and we had a covariate called effort. Effort here was how many days of the year a given interviewee spent in that unit. And again, so it's a random intercept, so it is drawn from a normal distribution of a certain mean and precision. For false positive probability, we had essentially just a covariate called 4+, which was essentially a binary variable indicating whether the number of interviews conducted per site was more or less than the mean. So the mean was 4 interviews in a site. So this is just to help the separation of the P11 and the P10. It was really assisted by a covariate on P10. And our thoughts were, we didn't have a great covariate in mind. However, we thought that at the site level, the more interviews that are occurring, the higher the probability that you're going to get a false positive in there. So we essentially just had the probability of a false positive as a function of the number of interviews within that unit. And it led to convergence, which was great, and the estimates made sense. So it worked for us for this particular exercise. So for probability of use, we used a logit link to model that probability as a linear function of covariates. And they had a random intercept of corridor to account for the fact that there are some corridors with a much higher or lower baseline probability of use than others. We had 6 covariates on psi. You can read as well as I can, but we have, cover, which included canopy cover and agriculture. We accounted for distance to settlement and elevation, distance to strictly protected area, and prey richness. So for our process model, our true occupancy state Z at site I was modeled as a Bernoulli random effect, which was a Bernoulli from psi. And yeah, the observed occupancy state Y at site I in survey J was also modeled as a Bernoulli random variable. And this was a function of apparent use, where apparent use is essentially a function of that true occupancy state, as well as your probabilities of true detection in P11 and false detection in P10. So that's connecting kind of the state process represented by Z with the observation process Y. So we collected about 4,000 interviews across 6 years in the 6 countries in an area totaling about 44,000 square kilometers. This was us completing that southern Belize corridor. No, this was the central Belize corridor, and we were very happy. And yeah, essentially the results here, holding effort constant, we found that P, which was probability of true detection, varied by corridor, field season, and year. There was a lot of heterogeneity happening here, so it's important to account for. But what was good was that there was no trend in year. So don't forget, like, this was a single season model conducted over 6 years. We were worried that with increasing species rarity, perhaps we'd see a decline in detection. We did not see that here. And so, yeah, it's going from left to right, going from the northwest all the way down to the southeast of Central America. And the colors are different years. So yeah, Jaguar habitat use was greatest with greater forest cover, greater distance away from settlements with more prey species, and it was less when you get more agriculture and pasture, when you're getting further away from strictly protected areas, and when you have higher elevation. And our biggest drivers here were percent forest cover and percent agriculture. And with the exception of the strictly protected area covariate, which had a 95% credible interval that just overlapped 0, the others did not overlap 0 at all. False positives were low and increased slightly with the number of interviews in a unit. So in units with greater than or equal to 4, we had a false positive probability around 8%, and it was more like 5.5 when a unit had less than 4. So that doesn't seem like a lot. It seems, okay, we're hovering around like 6% false positive rate estimated by this model, right? So that doesn't seem like a lot, but when you look at the actual estimates, this here is accounting for false positives. This is, again, probability of jaguar habitat use throughout Central America. This is accounting for it, this is not. Accounting for it and not. So failing to account for false positives biased our predicted habitat use high by around 2 times, 1.8 times. And again, that is a really dangerous thing when you're dealing with a threatened species such as jaguar. There are particularly large discrepancies for 2 corridors represented by corridors 4 and 10. These estimates were biased high 3 to 4 times. And when you look closer, you know, these the detection histories were largely all zeros, or there was a solitary 1. And so the model was basically saying, yeah, a lot of those solitary 1s are likely false positives. And then we had a couple different ways of validating our model. So bear with me, this is one of my more complicated slides. So don't forget, our observation process is a series of 1s and 0s reported by interviewees. There's false positives, there's false negatives. So we used models built with 80% of the data to predict Y, to predict that interviewee response in the remaining 20% of the data. So essentially, yeah, we essentially took out the Ys for 20% of our sites and made the model predict. And yeah, we had a means of, yeah, essentially if 1 was predicted for any survey, that site got a 1. And those values were compared to observed Y. And essentially what we got here, again, predicting those Ys to 20% of the data 5 times, we had a fairly good correspondence between our model predicted observations and actual. It wasn't perfect, so accuracy rate hovered around 0.65, sensitivity, which is the probability of reporting jaguar presence correctly, was about 0.69. And specificity, which is correctly classifying units without Jaguar presence, was around 0.6. So again, not a perfect model, but it does pretty darn well, because don't forget, this is a complicated process. Those Ys have true occupancy. It also has the false negatives and the false positives. There's a lot happening. So I was pleased with how this came out. In case that wasn't enough, we looked at a study by Wolch et al., published in 2016, revealing that genetic subdivision was most pronounced between Guatemala and Honduras, that there was a genetic, essentially, break between those two countries. Now here, that's represented by corridor 4. Okay, that's where Guatemala and Honduras meet. If we basically, the genetic structure observed in Wolch et al. Much better reflects our predicted habitat use by jaguars from our false positive model. As you can see here, predicted probability of use is extremely low there. However, had we ignored false positives, the picture looks a lot rosier, right? So this is our false positive corrected model. This is not corrected. And situation A is much more in line with those genetic findings. So our take-home points here are that you can get reliable predictions from interviews in spite of the data complexity. Again, we had our corridor-specific differences in baseline Jaguar occupancy, temporal differences in sampling intensity and duration, and we were still able to get some good estimates. And that interviews do represent an important yet underutilized source of data on species presence. But again, very important to account for false positives. Failing to do so can really inflate your estimates of probability of use or occupancy. So that concludes part 1 of 3. And I guess I can take questions at the end. Feel free to ask questions as I go. I don't mind. Question 2 was, how can the knowledge of local trackers be utilized to inform multi-species occurrence and guide anti-poaching efforts? And this work has been published in Animal Conservation. So we're going to hop on a jet and fly over to Angola, specifically Luangwa-Luana and Mavinga National Parks. And this area, in the dark green in the left, constitutes the largest contiguous national park complex within any one country in Africa. And so what makes this area even cooler is that if you look to the right, it is a part of this giant transfrontier conservation area called KAZA, K-A-Z-A. So this area is the largest transfrontier conservation area in the world. It is the size of Spain, and it has parts of 5 South African countries. It has Angola, Botswana, Namibia, Zambia, and Zimbabwe, and serves as a stronghold for lions, leopards, cheetahs, buffalo, wild dog, and elephant. So really important here to bring up the people who reside in and around the national park complex. They are the San people. San people, they are indigenous hunter-gatherers. They are the First Nations of Southern Africa, and their territories currently span multiple countries across Southern Africa. What they are perhaps most famous for is their, at least in the ecology community, is their tracking abilities. Their tracking abilities are considered legendary. When the Bushmen, when their track reading abilities have been tested against Western scientific standards, they have proven remarkably accurate and reliable. There's been work by Philip Stander on this, as well as recent work by Derek Keeping at the University of Alberta, showing how well these tracking estimates compare to other survey methods, if you're interested. And yeah, the use of San trackers just represents an exceptional opportunity to unify the advantages of a traditional skill set with modern quantitative methods. So that's where we're headed in this talk. Angola is a really, really fascinating area to work in. From 1975 to 2002, there was an Angolan civil war that led to the death of hundreds of thousands of Angolans, leading to the displacement of about 1/3 of the population. And yeah, people affected by war in general become increasingly reliant on natural resources, such as fuel wood and wildlife, to sustain their livelihoods, and they can seek refuge in protected areas for food and shelter. Within Luenge Luana and Mavinga national parks, wildlife was believed to have been slaughtered in mass during the war. This is a Panthera employee displaying a gin trap and a wire snare used to trap wildlife. So there is widespread bushmeat hunting and elephant poaching. And there are small villages common throughout both parks, particularly along rivers. So they, local people, they're hunting wildlife, they're fishing, they're grazing their livestock, collecting firewood, and there is poaching of high-value species such as elephant as well. There's subsistence slash and burn agriculture, which leads to the clearing of natural habitats. And, in general, the presence of people within protected areas can be detrimental to wildlife. And, there is often a lack of institutional capacity, and there's often a presence of funding shortfalls that make it really difficult to manage protected areas effectively. And that's been made even worse by the pandemic. So a really common survey technique on the African continent is vehicle-based spore transects. And so this is an efficient, low-cost method to detect multiple species across broad spatial extents. And they are considered to be more rapid and require less effort compared to camera traps. Paul Funston had a paper on that in 2010. What you need are suitable substrate, such as sandy soils, and sufficiently skilled trackers. And the San people absolutely fit the bill in terms of their tracking abilities. So here is our study area. The yellow cells were the ones that got surveyed. So each of those cells was 225 square kilometers. And this grid cell size was chosen to approximate the mean home range size of lions, which were the focal species of the survey, and to provide information on species distribution at a scale relevant to park managers. Originally, the goal was to survey everything, but turns out they were relying on roads and fire breaks, and they weren't present everywhere. And there were also areas that had not been cleaned of landmines. So there were some pretty serious safety concerns that had to be taken into consideration here. So the goal in each of those 225 kilometer grid cells or sampling units, the goal was to survey a transect of 15 kilometers in length. Every 500 meters, the trackers would record species detections, a 1 or 0 for each of those species of interest. There were 3 San people used as observers at a time. I think there were 2 sitting at the front of the vehicle, and the vehicle drives very, very, very slowly. And then, there was 1 who would cycle in every hour or so to reduce visual fatigue. And the surveys did have to pause between 11:30 and 2:30 because the sun was directly overhead, and it's impossible to see tracks when you don't have kind of some interesting light angles. So the trackers recorded tracks of all known large-bodied mammals in the area. There were 13. Moving from the top, and then I guess I'll go clockwise with the carnivores. We have leopard, we have hyena, wild dog, lion, and cheetah. And then moving clockwise with the herbivores, we have sable, rowan, elephant, buffalo, kudu, zebra, eland, and giraffe. The statistical challenges are that transect data are spatially dependent. So especially when animals are moving along a road or a fire break, if you see tracks of an animal in one 500-meter segment, you are certainly more likely to see it again in the next segment compared to a segment that did not have any tracks. And failing to account for this Markovian dependence can lead to substantial biases in occupancy estimates. Jim Hines had a paper in 2010 showing that. And community occupancy models had not yet been extended to this class of data. And community models in general allow for the estimation of both community level and species level effects. And that was of interest to park managers in terms of, how is the community doing, but also at a species level, where are they and what factors are associated with their presence. So our model was a hierarchical community-based occupancy model. And again, it incorporated that correlated detection model of Hines et al. And yeah, essentially, so you have a greater probability of detecting a species on a subsequent segment, given that it was detected on the previous. And it also had that space for time substitution. So you had your detection, non-detection replicates occurring in sequential spatial segments rather than in independent sampling periods. And there were two scales operating here. So, we do have our greater cells, but where the spatial dependence is occurring is at the level of that 500-meter segment. Model formulation here. So within the community occupancy model framework, we assume species level parameters represented by subscript K are random effects drawn from a normal distribution governed by community level hyperparameters. And each of those has a mean and associated variance. The top equation is our detection probability, and our bottom is our occupancy. Our beta represents our vector of coefficients estimated for D environmental drivers in a given grid cell I, and alpha and xi represent random intercept of species. So again, this is just setting up those hyperparameters for the estimation of both community level and species level effects. Okay, so the observed occupancy state, so the Y, so this is our data. These are the ones and zeros. Is the species there or not for a given segment? So that Y is at the level of each species K on transect segment J in grid cell I, and it was modeled as a Bernoulli random variable. And that Bernoulli random variable, so then the effective dot P from which the Bernoulli is drawn is the probability of a species being detected on a segment, given that it occurs in the cell. And that is a product of those two processes, your local occupancy and your detection. And so then your local occupancy is a function of whether the species K is present on the previous segment J and present in broader grid cell I. And so in that equation, you do have two theta terms. Each theta, there's one theta that is, the species was detected on the previous segment, or the species was not detected on the previous segment. And then the Z here represents true cell level occupancy, and it is a Bernoulli random effect of psi. So again, this is kind of connecting that state process and getting it back to our observations, which are the Y. So again, we have our occupancy at that broader grid cell, and we have our occupancy at the segment level. So it's integrating those two scales and bringing it back to our observed data. The covariates in our global model were split as either ecological or anthropogenic. So in parentheses, that's where we predicted the covariate would go when it comes to community occupancy. We had on the ecology side, we had our percent woody vegetation cover, we had precipitation and distance to water. On the anthropogenic side, we had distance to settlement, distance to other protected area, and human sign. Human sign was the average number of human sign per 500-meter transect segment. For results, there was widespread presence of people within the park complex. Human sign was observed in about 90% of the units, and livestock sign in about 80%. And bushmeat hunting was a pervasive threat. There was documented poaching of 19 mammal and 1 reptile species. Here are those 13 surveyed species, and in the numbers here represent the percentage of cells where the species were found. And you can see that for carnivore, we have leopard. For herbivore, we have sable and rowan were detected in the highest percentage of surveyed cells, so over 40%. And the zebra and kudu in the bottom right were detected in less than 5% of the grid cells and on less than 0.2% of transect segments. So essentially, those two species are presumed rare due to bushmeat poaching. So in the end, we excluded those two super rare species because we deemed the influence of those more data-rich species would pull estimates for the data-sparse species towards the community mean. And so we didn't think that that would be kind of a reliable species inference for those two. So we did exclude them ultimately from the community model, though the model would have been able to work with that data just fine We just didn't think it would really represent those species well. So these are our beta estimates for our covariates, and anthropogenic covariates in orange had much more explanatory value. Note that the 95% Bayesian credible intervals do not overlap 0. However, the biological ones absolutely did. And this was pretty unusual for me as a modeler, because I'd never quite seen anthropogenic covariates having such a massive effect compared to the more traditional ecological indicators. So again, looking at the prediction intervals here, there were much stronger slopes for those anthropogenic covariates along the top. The one that was really surprising to me was panel E, distance to water, because there are absolutely water-dependent ungulates in this system, namely elephant and buffalo. And it was counterintuitive to me, but then I remembered, wait a second, the majority of human settlements within our study area are along major rivers. So it could be kind of a competitive exclusion thing happening where, you know, the best riparian habitat is occupied by people. So here's where you get into the nitty-gritty of species level estimates. I guess I could talk about this figure for a long time, but what I'll do is just say that in general, again, those anthropogenic covariates at the top had a much stronger signal. Going from left to right, we had 8 out of 11 species, 11 out of 11, and then 7 out of 11, all with 95% CIs that did not overlap 0 in a given direction. Whereas with the biological covariates, it was just much different. There were just really mixed effects, and there really wasn't much of a signal through the noise. Species richness, so this is essentially just adding up the estimates for Z, that true occupancy state. Species richness ranged from 0 to 10.45 out of 11 species across the grid cells. And there was a clear decline in richness where human impact was greatest, notably in the northern half of the study area. And we threw the kudu and zebra back in just to see, you know, where those species were detected. Are those, you know, basically higher richness cells? And we did see that positive correlation. So we do think that those two species would follow the richness patterns that we see for the other 11. So then we have this richness map, and it's like, okay, well, that's really great. So you were able to use the vehicle-based spore transect data to generate this map. That's cool. But what has happened since this work has been done, the Angolan Ministry of Environment first is committed to tackling both the bushmeat issue as well. So difference between bushmeat and poaching. Bushmeat is considered largely subsistence or for small markets. Poaching is considered the killing of of high value species such as elephant. So weird like terminology thing. And they are committed to tackling this issue while partnering with conservation organizations. It has led to the identification of 2 intensive protection zones, each about 1,000 square kilometers, and both in that southeastern part of the complex. And there has been the creation of 22 jobs to protect wildlife and sustain local livelihoods within those IPZs. And those jobs include your typical game warden jobs and also community guardians to help communities essentially protect their livestock and themselves from coexisting with carnivores. So there's some good things happening. So take home points, you know, skilled trackers, you know, can serve as a high quality data source for use in a community occupancy model, and that the cessation of war is not adequate to counter wildlife declines if there are still people living within protected areas and when there's still enforcement challenges. However, I would definitely not end this section on a more depressing note, because, you know, with the assistance of conservation partners, it does absolutely seem that things are moving in a more positive direction and that wildlife recovery is indeed possible. And it was with the help of the SON trackers that we were able to prioritize, okay, where are the species? Where do we need to essentially lock down these core areas to conserve them? So moving, I can't tell if I'm not getting questions because I'm boring or because the talk is just so good. You have no questions, oh Lord. Just kidding. So question 3, can a community guardian program successfully deter livestock depredations by lions? And in addition, what factors are associated with short and long-term success? So our focal species here is the African lion, and it is vulnerable on the red list. Thank you, Stephanie. I can't see your full name. Vulnerable on the red list, and anywhere between 23,000 and 39,000 mature individuals remaining. And we're again gonna, maybe just take a cruise across southern Africa to get to Zimbabwe, specifically Hwange National Park, which covers about 15,000 square kilometers of semi-arid savanna in northwestern Zimbabwe. And if the figure on the right looks familiar, it's because Hwange National Park is present in the exact same transfrontier conservation area as Mavinga and Luenge Luana further west. So again, it's a part of this huge conservation landscape that is the size of Spain. And so the intactness of mammal communities is really important in order to promote dispersal of individuals and to preserve this greater metapopulation that exists here. Unfortunately, there is a lot of conflict between large carnivores and people in these areas. It's no different, I guess, from living in Washington State. And this conflict largely involves depredation of livestock. This is a global issue. When I was a PhD student, I did, the dreaded literature review of 178 peer-reviewed journal articles on human-carnivore conflict, and I found that it affects 37 carnivores across 6 continents. And essentially what happens is it leads to retaliatory killing and a perpetual cycle of conflict. So there are numerous approaches to reduce human-wildlife conflict. So this photo is an example of a non-lethal means. So this is just essentially a livestock corral made from local trees to keep livestock penned at night to prevent carnivores like hyena and lion and leopard from getting in. So there are, your lethal means, which is through targeted removal, hunting, and then, similar to this photo, there's the non-lethal. It can be through fencing, it can be through visual, auditory, chemical deterrence. It can be through a painful stimulus, such as pepper spray and rubber bullets. You just, in the case of the pain-inducing stimuli, you want to ensure that the stimulus is noxious enough to deter those unwanted behaviors. And I know in the literature, these types of stimuli have had mixed effects that, over time there can be habituation to the presentation of those stimuli. So where I'm going with this talk is, what about fear induction? Rather than, pelting lions with rubber bullets, what about just getting them to be really afraid, particularly of people, without killing them? And so that is a tough thing to do, but that was what we were trying to do with, I should say, what they were trying to do with this project. I cannot stake any ownership in this project. So let me introduce you here to the Hwange Long Shields Community Guardians. These men and women, they are a mix of, they're mostly Ndebele people. They speak Ndebele, they speak Nambia and other regional African languages. And in the past, 19th century Ndebele, they would have hunted and killed lions as the Maasai do today. However, such practices stopped with the advent of the Zimbabwean protected areas system. Of course, it still occurs, but it's frowned upon and they could be fined and jailed, etc. So what villages do now are they keep lions and other carnivores away by lighting fires, by banging pots and pans, essentially creating a ruckus to say, hey, do not come here. This is a dangerous place. So Wild Crew, which is the Wildlife Conservation Research Unit at Oxford University, took it a step further and spearheaded a program that essentially relies on those types of principles, but hires local people to essentially monitor lion movements, warn people when they come close, and most importantly, they will chase them. So these Long Shields guardians will physically chase lions away when they come too close to community lands. And a lot of the lions in the system are uncollared, but some of them, at least one lion from each pride, does have a GPS collar. So these guardians have the additional knowledge of live tracking from GPS collared animals. And when a lion gets too close to their village, they will get a text message, they will get on their bike, and they will essentially chase after a lion. And yeah, the lions are chased on foot or by bicycle. They are chased with these plastic horns that make a really loud, terrible sound. And the goal is for lions to recognize community lands as high risk and to shift their space use to include more of the park. So since the program started it was up in earnest in 2012, but they were kind of operational in 2011. The number of depredation events in both community areas had decreased as a result of the program. So less livestock were being killed, which is really great. In addition, less lions were being killed. So it seems like this program is actually like kind of working here. Yeah, so there's multiple ways lions can yeah, they are indeed vuvuzelas. Yeah. Yeah. So, yeah, there are 3 ways that lions can be taken out. They can be taken out through problem animal control, which is when Zimbabwean parks and wildlife will come in and lethally remove a problem animal. Communities will go after the lions themselves, though I have heard of community members being killed as a result of that choice. And illegal poaching is snaring. It's often not meant for lion. Sometimes it's meant for ungulates, but lions get involved and yeah, they die as a result of being snared. So this is a pretty short section here. Yeah, so my question was, what factors are associated with immediate chase success? So these Ndebele people are chasing after these lions. What factors are associated with a successful chase, meaning the lion is pushed out of where they're not supposed to be by 7 a.m. the next day? And in addition, I was wondering, okay, what about these longer term behavioral changes by individual lions? What factors are associated with that? So I was thinking of these 2 different time scales. So we did use GPS collar data here. So there were a total of 109 lions collared within the study area over the last 18 or so years. However, only 15 met our criteria. So they had to be from unique prides and coalitions. They had to have part of their home range overlapping community lands. And we had a mix of adult males, adult females, and subadult males. Why there were no subadult females, I couldn't tell you. They were monitored for an average of a year, and each lion was chased between 0 and 17 times. So quite a wealth of data to work through here. So here is the study area. The protected area is in dark green. The national park, I should say, is in dark green. The forestry lands where people are allowed to take firewood and things is in the light green. And then the hatched area is like community lands. The red points are villages. So like once lions step close, I think within 2 kilometers of the hatched area, the guardians would get a text message and they went to work chasing after these lions. So the methods here for chase success, it's the most basic analysis I think I've done in the last 10 years. It was a mixed effects logistic regression using individual lion as a random effect. And for the longer term behaviors, so we were looking at the behavior of entering community lands, which was bad, and also the behavior of depredating livestock, which is bad. These behaviors were analyzed separately, and they were analyzed using recurrent event, gap time, Cox proportional hazards models, which is a mouthful. What those models do, for those of you who haven't used them, they allow you to examine how certain factors influence the rate or the hazard rate of a particular event happening at a particular point in time. And a lot of times you'll see Cox models used for survival, so whether an individual lives or dies. In this case, because it's a recurrent event model, you can incorporate multiple events per subject. So a lion will enter community lands and then enter again and then enter again. Same with depredating livestock. So it just allows for that hazard to be estimated over multiple events rather than just a single death. We have our hazard function here, which is the risk of an event occurring at a certain time t, and it's determined by a set of p covariates. And that is within our model statement that I indicate here. And as well, we have a baseline hazard, which essentially corresponds to the hazard if all the x's are 0. And this is an intercept term that varies with time. It is not estimated by the model. However, a really important assumption here is the proportional hazards assumption. It is assumed that the ratio of the hazards for any two individuals is constant over time. And you need to run through a series of tests to ensure that your model, that your data do uphold the proportional hazards assumption. Okay, so we had a bunch of covariates. I had 25 of them. I won't bore you with what all of them were, but I did group them into 5 categories. We had our early intervention. And again, these were the covariates that I thought would be associated with both chase success and the occurrence of those 2 behaviors we don't like. And so we had early intervention. So maybe when you chase a lion early in its sequence of depredations, that's a positive thing. To the contrary, if a lion in the positive reinforcement category, if a lion has already killed 20 livestock, maybe it doesn't matter at all whether or not you chase it because of that positive reinforcement of having a successful livestock kill. Then we have negative reinforcement, which is that chase intervention. If it was chased on the previous entry, how does that affect the rate of having another undesired behavior? The social resistance category was arguably the most interesting to me. So this is your loss of a pride male in the last 6 months. So when you lose a pride male in the system, it's socially disruptive, and we'll see what happens. And then something like temperature for environmental resistance. Lions don't want to move when it's hot. So in general, 56% of the chases were successful. Lions had gone back out of the exclusion zone by 7 a.m. the next day. 44, obviously, were not. They were less successful over time. After chase 10, success was around 30%, as opposed to 60% after your first chase. So the more of these lions are being chased, the less successful they are. I'll go through these quickly because maybe they're common sense. If a lion is closer to the boundary of the national park, they're easier to chase. You're going to have a real easy time chasing them 200 meters. When you have to chase them 25 kilometers, it gets a little different. I have to make a herding cats joke here because we are literally herding cats here. When you're trying to get 1 or 2 to do a certain thing, it's a lot easier than trying to get 9 or 10 lions to do what you want them to do. For the interest of time, I might skip the season panel and talk about the pride and stability panel. So essentially, when there was pride instability, when there had been the loss of a male, which your success rate in chasing a lion out of the exclusion zone really decreases. And that was not surprising. When you remove a pride male, it causes a lot of social upheaval. There's competition among neighboring males for pride ownership. There's the eviction of subadults not yet at sexual maturity. There's infanticide of female cubs. So I can see why certain individuals would seek refuge near livestock and communities and not go back into the park where there could be a dominant male who would kill them or their cubs. So adult females and subadult males were misbehaving more so than adult males. They were much more likely to enter the exclusion zone. And it was just related to age class here and sex. Nothing else really came out as important. So only 10% of re-entries to the exclusion zone were followed by a chase. So this is clearly room for the program to improve. And yeah, 46% of those re-entries not followed by a chase occurred at night. So a lot of these re-entries aren't being responded to. And a lot of these are happening at night when people are sleeping. And I'm pretty sure we don't want the guardians to go chasing after lions at midnight. So in general, this slide here, again, I do want to leave time for questions. The more you reinforce, so let's just say when lions are chased 40% of the time after depredating livestock, that's in green, you can see the probability of another depredation is pretty low. However, if you only chase them 10% of the time, there's a high probability of a repeat depredation occurring. In general here, the more animals a lion is killing, the more chance that that lion will depredate again. So repeat livestock killers here killed an average of 29 animals for a maximum of 56 animals killed by those repeat. But no, I'm sorry, that was a maximum of 56 by a single individual. So some of these lions are absolutely chronic predators of livestock, and perhaps no amount of chasing can help. So generally, employment of local residents can promote tolerance of large carnivores. This program is Zimbabwean run. It boosts the economy, it builds capacity, also a sense of ownership in wildlife management. And that, yes, attention to social context and as well as consistent intervention are both paramount for the success of programs like these. And of course, room for improvement, but some really promising results we're showing here in essentially putting management of wildlife into the community's hands. So this is a sign from Belize, and this is my closing slide just to say that, yeah, this is the conclusion, I guess, of your whirlwind tour of how harnessing the power of communities and their knowledge can guide conservation and management efforts. So thank you seriously so much for inviting me to be a part of this seminar series. I'm pretty sure it's different from the previous ones. I'm sorry that I'm not a marine scientist and that I'm not a statistician, but I think I'm pretty close. So yeah, I'd really like any questions that you have.
Thank you, Lisanne. That was a great talk. Give folks a chance to fill in some questions here. And okay. When you do get a question, could you please read it back to the audience?
Yes, so I already see it. How did you account for, okay, so I will need, so I was asked, how did I account for detection heterogeneity on my occupancy model? And I'm guessing, thank you, Trevor, I'm guessing that is the community occupancy model. And that is because I didn't talk about it in the interest of time. So that is a great question. Where would I have, okay, so yeah, these were the, I'm guessing it's the community model. So these were the covariates on occupancy and on detection. I'm sorry, I didn't mention this. I had both transect length because some transects failed to reach the full length if they were at the very end. And also the number of human sign were on detection. And not surprisingly, there was greater detection with longer transect length and also less detection, less probability of detection with greater human sign. So thank you for asking about that.
Wait a few moments here to see if we get some more questions. Let's see here.
And if you're not the type to like asking questions and these types of things, feel free to find me either on Twitter or through email. I'm happy to answer questions there.
I think Laura James had a question. I'm trying to unmute her.
Hi, Lisanne. Thanks for your presentation. It's really great. I just want to know if you consider the effect of the road when you were counting the tracks of the animals, because for example the tracks of the lions the rate was very low, and maybe it could be because of the road effect or something like that.
Yeah, so I should mention that all of the transects were done on the same type of substrate. So within that national park complex, it's a lot of sandy substrate. It's not like super hard soil. However, what I did say, because there were differences in substrate quality, and I believe the trackers rated that quality from 1 through 4. And it's funny, when I put the substrate quality on detection, it had no effect at all. Yeah, I couldn't help for the fact that the transects were largely conducted on roads, but I did try to account for quality, and there was just no effect.
Okay, thank you.
No problem. I did not. So sorry, I should say what the question is. Michelle asked, did you account for spatial correlation between grid cells in the community study, or only correlation along the transects? And I only accounted for that correlation at the transect segment level, not at the level of the grid cells. But that's a really good thought. And now that I'm I kind of wish I had, but I guess that ship has sailed, but that's a great point. Another question, we're at 4:30, but it's a quick one. How many strikes before a bold animal is sacrificed? And in some cases, they do not allow strikes. If an animal has killed livestock, it can be taken. I wouldn't say there's an official policy on that. And if it's not taken out through problem animal control, local people may just do it for them. So, yeah.
All right. Well, thank you, Lisanne. That's all the time we have for today. Thanks for the great talk, and thank